Welcome to the Explainer. Today, we're grabbing our magnifying glasses and becoming genetic detectives. We're on a mission to hunt for clues hidden deep inside the code of life itself, our DNA. So let's just think about this for a second. Imagine you need to find one specific house somewhere in an entire country. But here's the catch. You don't have a map, there are no street signs, not even an address. Well, that's pretty much the challenge scientists face when they try to navigate the massive, unmapped landscape of a genome. So, how in the world do they solve it? Well, they build a totally different kind of map. Instead of being based on physical miles, this map is all about relationships and travel patterns. This is the story of genetic linkage mapping, a really clever technique that turns family inheritance into a working blueprint of our DNA. All right, so every good detective story has to start with a fundamental clue, right? For us, that clue is a concept called linkage. Okay, I want you to picture a chromosome as a really long bus, and all the genes are passengers on that bus. Genetic linkage is a simple but super powerful idea. Passengers who are sitting right next to each other are very, very likely to get off at the same stop. They're basically travel buddies, sticking together on the journey into the next generation. So really, it all just boils down to how close they are to each other. Linked genes? They're close neighbors on that chromosomal bus, so they almost always travel as a pack. Unlinked genes, on the other hand, are either on completely different buses, meaning different chromosomes, or they're sitting so far apart on the same one that they just go their separate ways. Okay, so if genes are traveling companions, how do we measure the distance between them? Well, the trick is to look for those rare moments when they actually get separated. That separation happens during this amazing genetic shuffle called crossover. It's where chromosomes literally swap pieces of themselves during meiosis. The frequency, how often a crossover event splits up two specific genes, is our measurement. Think of it like a key piece of forensic evidence. If two genes get separated all the time, they must be far apart. But if they're almost never separated, we know they're sitting right next to each other. And we measure this genetic distance in a special unit called a Centimorgan, or cm for short. And it's actually pretty simple. 1 centimorgan equals a 1% chance that a crossover will happen between two genes. This is the fundamental unit of our genetic map. So, how do we get from this one measurement to a full map? Well, by calculating these recombination frequencies between lots and lots of genes, we can start to place them in order, figure out their relative distances, and slowly piece together the entire blueprint. And this brings us to a really, really important distinction. The genetic map we've been talking about, it tells us the relative order of genes based on how they travel together. But there's also a physical map, which is more like a satellite view. It tells us the precise physical address measured in actual base pairs. You can think of it like a travel itinerary versus a specific GPS coordinate. But here's the kicker. Neither map tells the whole story by itself. It's only when you overlay the genetic itinerary onto the physical GPS map that scientists get the complete picture, not just where the genes are, but how they actually behave and interact when they're passed down. You know, this all might seem a little abstract, so let's see this detective work in action with a real-world case, the hunt for the gene that makes radishes red. So here's how the investigation went down. First, they crossed red and white radishes. Then they sequenced the entire genomes of 137 of the offspring. This created a colossal amount of data, with over 400,000 genetic markers, these little signposts in the DNA to track. By meticulously calculating the recombination frequencies between all those signposts, they built an incredibly detailed genetic map. This let them do something called QTL mapping, which is just a powerful way to link a specific trait, like color, to a specific spot on a chromosome. And guess what? The map worked perfectly. With this new guide, they scanned the entire radish genome and traced that red skin trait to one very specific neighborhood. The culprit? A single gene, which they called RSMYB90, right there on chromosome 7. But finding the gene's location wasn't the end of the story. It was the key that unlocked its function. They figured out that RSMYB90 acts like a master switch for making anthocyanin, which is the very pigment that gives radishes their bright red color. Case closed. Finding the gene for a red radish is fascinating, sure, but what does this powerful tool really mean for us, beyond the grocery store? Well, the impact of this kind of mapping is just huge. In agriculture, it helps us breed crops that are more resistant to disease or that can produce more food. In medicine, it's one of our best tools for hunting down the genetic culprits behind inherited diseases like Alzheimer's. It's even helping us decode our own evolutionary story and the very basic functions of life's code.
At the end of the day, genetic linkage mapping does something truly remarkable. It translates the abstract code of DNA into a functional story, a story about our traits, our health, and our history. It gives us the blueprint to really understand the genetic basis of who we are. The first maps have been drawn, and the tools we have are sharper than ever before. So as these maps get more and more precise, the only question left is, what incredible mysteries hidden deep in our own DNA are we going to solve next? The age of genetic exploration has really only just begun.